So yeah. No. Yeah, there's 14. Okay. Outlook's telling me not to forget about my seminar.
I thought it was mine. I was turning mine off. More.
Let me know when you think it's good to go.
I told Lois to start whenever she likes.
Yep.
Okay.
Okay, so I'm going to try and come into view here on the computer. So it's a great pleasure today to welcome Tim Walker to give his first seminar as a PhD student. So Tim is a PhD student in my lab over in Botterill Hall. More than Tim began in our lab back in 2019. He was a summer student and was a SWEP student with us, and he was one of the most excited students to actually get involved in research that we've ever had in the lab, which is a lot because we've had some very excited people in the lab. And Tim's was one of the first major cohorts of BSc, MSc students that came through pathology and had the good fortune to be in the middle of that when COVID hit, and he had to take a sabbatical for a few months in the middle of that, but he's transferred into his PhD now. He's been phenomenally productive, and Tim probably won't tell you, he's an author of a cancer cell paper that was submitted last week. Work he's going to talk to you today about is work that comes from his own hands, which we hope is going to be submitted in the next few weeks. So the timing for his seminar is perfect. So without further ado, tell us all about it.
All right. So thank you everybody for coming. We've changed the title since we sent out that seminar notice. So now the title of my project is Loss of Tumor Suppressor TMM127 Drives RET-Mediated Transformation Through Disrupted Membrane Dynamics. So that's a mouthful, and it doesn't really tell you what we're working in. So I'll give you a little bit of background on the disease model we're focused on. So we're interested in pheochromocytomas, which I'm going to denote as PCCs going forward. And these are rare neuroendocrine tumors that originate in the adrenal medulla. It's making noise. Peter, could you turn your microphone off, please? Thank you. So there are rare neuroendocrine tumors that originate in the adrenal medulla. Most of these tumors are benign. But because of increased release of catecholamines, these tumors often present with symptoms of kind of headaches, sweating, hypertension, and palpitations. And these can eventually progress to cardiac arrest or death. Most of these tumors, since they're benign, they primarily use proliferation. They're not very motile. However, a small subset, about 10%, actually are metastatic, with 5 and 10-year survivals of 65% and 35% respectively. Additionally, these tumors have very limited treatment options. Generally, the first line is surgery, and there's very kind of limited ways to treat these tumors. Additionally, of all these cases, about 70% present with either somatic or germline mutations in PCC susceptibility genes. And these susceptibility genes are clustered into groups of genes and 2 proteins of interest that are within the kinase signaling cluster include rearranged ring transfection, or RET, and transmembrane protein 127, or TMEM127, indicated in this diagram here. So for a little bit of background on these two proteins, so we can start with RET, or rearranged ring transfection. RET is a well-characterized proto-oncogene. It's also a growth factor receptor, which is required for the development of the kidneys and nervous system. And it generally kind of turns off when you're an adult, for the most part. However, RET contributes to cancers through a variety of different mechanisms, including constitutively active RET mutations, which drive MEN tumor syndromes, which are characterized by medullary thyroid carcinomas and pheochromocytomas. But also wild-type expression has been observed where RET is not actually the driver, but increased expression is seen. And these include some pheochromocytomas. Now for a more technical look at RET. RET is a single-pass receptor tyrosine kinase. It's made as an immature protein at 155 kilodaltons. It moves through the Golgi where it's post-translationally glycosylated to 175 kilodaltons, which is the mature protein. And therefore it shows up as a doublet Western blots, the immature and mature protein. Once protein is mature, it moves to the cell membrane where it can be activated in the presence of its ligand, GDNF, and its co-receptor, GFR alpha. And upon activation, RET can signal through several signaling pathways, including PI3K and RAS-MAPK pathways. And these stimulate several cellular processes, including cell proliferation, migration, and survival. And some proteins of note that will come later, ERK, as part of the RAS-MAPK pathway, as well as AKT and S6 as a substrate of mTOR, which are part of the PI3K pathway. So following activation, RET is internalized in the cell. This internalization is essential and very important in the regulation of RET signaling. So RET's brought into the cell where it can be phosphorylated and recycled through the endolysosomal system. So it first enters the early endosomes, and then it can be recycled back to the cell membrane for further activation, or progress to late endosomes and then down to lysosomes for eventual degradation. And now coming to the other protein, we're interested in transmembrane protein 127. It's a poorly understood tumor suppressor protein. It's broadly expressed at low levels in normal tissues, and as you can see in the diagram, it's a multi-pass membrane. It's an integral membrane protein, which has roles in endosomal transition and cargo trafficking. So it associates with the membrane and has some suggested role in endosomal transition, but again, it's not very clear so far. TMEM mutations, on the other hand, show consistent loss of the wild-type allele, consistent with being a tumor suppressor. And these proteins become mislocalized from the membrane. They kind of are unable to associate with membranes, so they're mislocalized. And we also see an increase in mTOR signal as part of the PI3 path. So all that taken together, our rationale behind this project is we know that TMEM inactivating mutations, that these mutations denoted just with the strike through here. So TMEM inactivating mutations can lead to pheochromocytomas. We know that constitutively active RET mutants also drive pheochromocytomas. We have also seen, or not we, but literature says that increased wild-type RET has also been seen in some pheochromocytomas. However, the role of wild-type RET in these pheochromocytomas with mutations in other susceptibility genes has not been investigated. So given that trafficking is critical to RET regulation and TMEM has suggested role in this protein trafficking, we predicted that TMEM loss could alter RET trafficking and function. So we thought it was something more along the lines of we have mutated TMEM, which is affecting RET in some way, which drives the pheochromocytomas. And to investigate this, our first question was basically, where is TMEM, and how do TMEM mutations affect where TMEM is? And we did this in a transfection model. So we have our PE cells transfected with either wild-type TMEM or two mutant TMEMs, C140Y or S147 deletion. And here I have the membranes internalizing. So as the first step, host membrane to endosome, we wanted to see if TMEM mutation affects localization, the step of internalization. And what we have, so I have the images here and a graphical depiction of the co-localization here. So here, these yellow puncta indicate areas of green and or TMEM and EA1 overlocalization. And it's also shown here, so there's quite noticeable localization, both relative to TMEM and relative to EA1. But when you use a mutated TMEM, you see a significant reduction in the amount of co-localized or yellow puncta. So we see a significant drop in co-localization. Additionally, up here, I've included the number of TMEM puncta and EA1 puncta per cell. So as we would expect from the mutant TMEM, they're unstable, so we get kind of less TMEM puncta per cell. But interestingly, in the number of EA1 endosomes, we see a moderate decrease with the S147 deletion and a significant decrease in the number of EA1 in the C140Y TMEM. So that could kind of indicate that TMEM has some type of information of endosomes, which we may get back to a bit later. So all this in mind, we wanted to kind of see how RET plays into this process. So we did the same thing, however, this time labeled for RET as well. So again, we have the wild-type TMEM and 2 mutants. And with the wild-type TMEM, we see, as indicated by these white arrows, the co-localization of EA1, TMEM, and RET together in these early endosomes, as we would expect. However, when TMEM is mutated, we see a loss of the triple co-localization of TMEM and EA1 and RET. So TMEM kind of pops out of these endosomes and no longer localizes with RET and EA1. RET there's some splotches of purple, which would indicate some RET and EA1 co-localizations still occurring, so it's not eliminated, but it could also be reduced. It's tough to tell based on these images, but so these 2 experiments together, transfection models are they're great to assess many things. However, when dealing with a system as sensitive to protein overexpression, protein endosomal trafficking, we thought that an endosomal or an endogenous model would be more biologically relevant. And given the consistent loss of the wild-type allele shown when TMEM is mutated and the loss of integral membrane protein function of the mutants, we went forward with an endogenous knockout model to become biologically relevant. And this is what we did. So our endogenous model are SHSY5Y neuroblastoma cells. We have our mock knockout cells, which should express RET and TMEM. And then we have a knockout version, so we can have a case where TMEM is knocked out and see the effects on RET. And in the process of kind of characterizing these cells, I just looked at kind of simple protein, a Western blot to see the protein expression. So in the mock knockout cells, we see RET and we see TMEM, as we would expect. And then TMEM knockout cells, we lose this expression of RET. But interestingly, we saw a large amount of RET, large amount of mature RET present here. And given this is the mature protein, which is generally on the cell membrane, we were curious to see if this accumulation was occurring on the cell membrane. And to do so, we did a simple surface biotin lation assay. So with a biotin conjugate, we're to label kind of all the cell surface proteins as biotin. And then we were able to immunoprecipitate with streptavidin, which has a high affinity for biotin. So any of the proteins pulled down are labeled with biotin, meaning that they were on the cell surface. So I've shown here the precipitations, these 2 top plots. So TMEM is on the cell surface, as we would expect, as is RET. However, in the TMEM knockout cells, RET is quite noticeably increased over the mock knockout cells, and I've also included a graphical depiction here. So we see about a 5.1-fold increase to total RET protein levels on the cell surface, which is interesting. Yeah, so we lose TMEM and we get an accumulation on the cell surface. So our first question was, is there a default in protein internalization? Because if proteins weren't internalizing properly, stuck on the cell surface. So that's exactly what we looked at. We used a slightly more complex biotinylation assay. So again, we label all the cell surface proteins with biotin, and then we would treat with ligand for 5 or 30 minutes to allow for internalization. And then we could strip the surface of any remaining biotin-labeled proteins. So the only biotin-labeled proteins remaining are the fraction that have been internalized. So I did this in my mock knockout and TMEM knockout cells. So again, we have 100% all the biotin-labeled proteins, and then the fraction about 9% after 5 minutes and 28% after 30 minutes in the mock knockout cells. But contrastingly, in the TMEM knockout cells, we see higher internalization by 5 minutes, about 16%, but a non-significant increase over 30 minutes, going from 16 to 18%. So this to us suggested that there's less robust internalization overall, but also there's kind of some level of background internalization that's insensitive to GGNAF. So this was interesting. So you lose TMEM, get less internalization, it's getting stuck on the cell surface. But in order to figure out exactly kind of why this is occurring, we wanted to look more specifically at the actual process of RET internalization, which takes me to a little bit more background. So the RET internalizes through clathrin-mediated endocytosis. And this process, basically, when RET is activated, it recruits clathrin-associated adaptor protein 2, or AP2, which then recruits clathrin, which kind of builds a lattice around this forming pit. And that's kind of depicted here in this image of, we're kind of looking at the bottom of the membrane. So as this clathrin lattice forms, it kind of starts off flat, and coalesces together in this lattice structure. And then as it builds, it kind of gets curvature and pulls away from the membrane. So this is kind of a more pulled out view of that. And then once you get to kind of a fully formed vesicle, also known as clathrin-coated vesicle, eventually it will pull away and be cleaved off the membrane. It can now travel into the cell. The clathrin lattice will fall apart, and then this vesicle can enter the endolysosomal system. And so since all of this is occurring right at the cell membrane, it's fairly tough to do this kind of look at these kind of proteins in a confocal or epifluorescence type of microscopy. So what we used is a specialized type of microscopy, total internal reflection fluorescence microscopy. So with your epifluorescence, you shoot the laser straight through the cell and excite the fluorescent proteins in the whole 3D structure of the cell. However, with TIRF, the laser is kind of shot at an incident angle where you get total internal reflection. So you only excite the proteins right at the cell membrane, right at the bottom, through this evanescence field excitation. And this only penetrates about 90 nanometers into the cell. So again, you kind of everything on the cell surface, everywhere the cell is stuck to the glass. And so this is exactly what I did. So I looked at starting off with some fixed cell TIRF. So these cells were power slips. I have my mock knockout cells and TMEM knockout cells in the presence and absence of GDNF. And I've labeled for RET and clathrin. And then I have the merged channel here with clathrin and any areas of causation indicated as yellow circles. I've also shown this graphically over here. So in our mock knockout cells, in the absence of GDNF, we see some level of baseline co-localization. And when we add GDNF, as we would expect, RET gets localized to these clathrin-coated pits. It's forming areas of clathrin it will internalize. Again, there's a part of the diagram again over here. However, in the TMEM knockout cells, again, we don't see a significant increase without GDNF, but upon GDNF, we see no significant increase in the amount of co-localization. So ultimately, that said to us that there was reduced RET clathrin co-localization on the cell membrane. And it is also insensitive to GDNF, which kind of cropped up earlier. This idea that these cells are not responding. Additionally, from these images, I extracted some more information. So here I've shown the intensity of clathrin within CCPs. So in the TMEM knockout cells, you can see there's a significant decrease in intensity clathrin with CCPs, suggesting that they're potentially smaller and have less clathrin in them. Additionally, along with the idea that there's more red on the cell surface, I see higher red intensity in the TMEM knockout cells. But interestingly, I did the same experiment and I labeled for EGFR, and I saw a comparable increase in the intensity on the cell surface, or increase in the intensity, suggesting that this is not only RET specific, it's more of a global effect, which would be very interesting. We had no reason to suspect it was RET alone that was being affected. And the fact that we see a comparable amount with another receptor that traffics in the same way through cytosis, we thought that was very interesting. So it's a global effect, and potentially this protein was changing membrane dynamics or something, not allowing for internalization. So to examine the effect of TMEM loss on these membranes without considering RET, we used live cell imaging, again with TIRF, so everything up the cell membrane. So these are my same TMEM knockout cells, but they express a fluorescent clathrin. And these videos were taken over 5 minutes, an image taken every second. And I put it's 15 seconds. So before we even quantify, I thought it was very interesting. I hadn't looked at them when I was putting all these things into our process of quantification. But when I made these GIFs, I was like, these little bits of clathrin knockout cells, they seem to be jiggling around quite a bit. They're very kind of motile, which as we would expect. And kind of, because it's everything at the membrane, as things come close to the membrane, they appear, they jiggle around and then disappear as they away from them. On the other hand, the TMEM knockout cells, by comparison, there's still some coming to the membrane and leaving, but overall, it seems very stagnant, to me at least. Everything seems kind of stagnant and not jiggling around as much. So that was before even quantifying. So before I get to the graphs of quantification, I wanted to explain a little bit more about clathrin cytosis. Everything in science is more complicated than you think, so I started this again. So in the process of forming these clathrin-coated pits, there's multiple stages. It's a very complex process which requires multiple kind of stabilization steps. So the initial step, you get AP2, comes to the membrane, and it kind of recruits clathrin to kind of nucleate this process of initiation. So you get a little nucleation that goes on and would become initiation. However, sometimes this is unsuccessful, and these little clusters don't recruit enough clathrin to become a CCP. So that's what we denote as subthreshold clathrin-labeled structures. So they're little clusters of clathrin that eventually fall apart and don't go to initiate a CCP. But if they do, then clathrin-coated pit initiation starts, and then there's multiple maturation processes after that. So you can get initiation, and then it eventually goes and makes a productive CCP. But it can also fall apart after you initiate it, and it can fall apart before it becomes productive. We call it CCPs. So again, it's a very complex process, but hopefully this diagram helps with these graphs. So this first graph, I've shown the nucleation density, clathrin-labeled structures that don't go on to form CCPs. So in the TMEM knockout cells, we see a significant increase in the amount of these little clusters that fall apart and don't form CCPs, which then results in a decrease in the amount of actual CCP initiation, as we would expect. However, even though there are less CCPs, they are more productive. So the fraction of CCPs less than 15 seconds, which are the abortive CCPs, we see a decrease in that fraction, so they're actually more productive. However, there's less of them. And also, going along with pathogen intensity from the last slide, they're also potentially smaller. So lots of little clusters that fall apart. Some are able to form CCPs. They're smaller, but there are more. They're smaller. And that would make sense with the internalization, because they're not to potentially grab as much RET or recruit RET to internalize compared to cells. So this to us suggested that TMEM played maybe some type of role in stabilizing them to be able to form these Clathrin pits. And again, this is an area where we have to do some more investigating to figure out exactly kind of what stage this loss of TMEM is affecting. But I'll come back to that in our future directions. But now going back to kind of the concept of how this affects RET. RET's kind of stuck on the cell surface. It's not internalizing. We would expect to see a decrease in the amount of RET degradation. So cyclohexamide degradation assay. So cyclohexamide stops protein synthesis. So you kind of have a starting amount of protein. You're not making any more, and you watch how it degrades over time. So in our mock knockout cells, we start with 100% of RET, and then kind of over time, we end up with about 9% hours. So it's quite a significant decrease over time. However, in the TMEM knockout cells, we start with 100 again, and we have about 43% remaining after 12 hours, which is a significant decrease in RET degradation and an kind of increase in RET. Because it's getting stuck at the cell surface, less it's entering the cell and able to go down to the lysosomes. And kind of with all the RET on the cell surface, it's kind of simulating an overexpression because RET isn't able to be brought in and degraded. And we know that kind of overexpression of receptor tyrosine kinases can lead to constitutive activation of receptors. And so I was curious to see if this accumulation is leading to constitutive activation. And it seems like it is. So here I have Western blots probing for phospho-RET, phospho-AKT, phospho-ERK, and 6. So I have my mock knockout cells and TMEM cells here, and I have a graphical function over here. So ultimately, in the mock knockout cells, you see very minimal activation in without GDNF. Compared to the mock knockout or the TMEM knockout cells, we see noticeable phosphorylation of RET, AKT, ERK, and S6. S6 again is a substrate of mTOR, which we know loss of TMEM increases mTOR. So there it is right there. And also, yeah, if we look at these graphs, so we see a significant increase in constitutive activation, but also when adding GDNF, the mock knockout cells respond as we would expect, kind of very minimal activation to turning it all on. However, the TMEM knockout cells don't show a significant increase in activation when you add GDNF. Again, GDNF insensitivity that's been cropping up there. Interestingly, the case with RET, so there's a non-significant increase in the amount of constitutive RET, constitutive RET, but because there's so much RET, it shows up as an increase on the Western blot. But also, it's increased in the presence of GDNF, but insensitive to longer treatments. Again, it doesn't change over the half an hour as much as the mock knockouts do. They kind of consistently increase. So we thought this was very interesting. And next, we wanted to see kind of if we could knock back this constitutive activity RET inhibitor. So here, vandetanib is a multikinase inhibitor, and we have sulfatinib, pralsetinib, which are both RET selective inhibitors. And in the presence of all three of them, we were able to knock back this constitutive activity, showing that it was RET mediated, and we were able to so and we know that many of these signaling proteins are involved in signaling pathways that contribute to cell proliferation, and that's kind of the primary driver of pheochromocytomas. So we wanted to kind of see if this constitutive activation was leading to an increase in cell proliferation. So to do this, we used an MTT cell viability assay, so we can measure cell viability over 3 days, and we see that the TMEM knockout cells, the mock knockout cells in the absence of GDF, they still grow, but very minimally, and we see an increase in growth when we add GDNF, as we would expect. However, in the TMEM knockout cells, we see significant growth with or without GDNF. So these cells don't care if you add its ligand. The constitutive activity is enough to sustain their accelerated proliferation. And again, since inhibitors were so effective, we wanted to see how inhibitors would alter this proliferation. So again, we see the high growth in the TMEM knockout cells, less so in the mock knockout cells. But then when we add selpercatinib, we could kind of stall cells, keep it at one-fold. However, in the TMEM knockout cells, we not only knock back that proliferation, we actually lead to a 19% reduction in cell viability, suggesting that these cells are potentially sensitive to RET inhibition. So that was a lot of information. I've summarized it in this diagram, which is hopefully helpful. So in cases where you have wild-type TMEM, you get kind of proper formation of these big CCPs. They bring in RET and TMEM, and then go down to degradation. There's a lot of them, and they're fairly large by comparison. In the TMEM knockout cells, we get an accumulation on the cell surface. This leads to some constitutive activity and an increase in signaling, which ends up in an increase in proliferation. We also see a decrease in the number of CCPs, in the size of the CCPs, and a decrease in RET to these CCPs. Additionally, from the transfection model, we see a decrease in the number of early endosomes, which could go along with decreased size of CCPs. We see reduced RET internalization and decrease in RET degradation. Additionally, when we add a RET inhibitor to these cells, we get an increased sensitivity and a resultant decrease in cell viability. So ultimately, this kind of RET mislocalization could be the pathogenic mechanism underlying these RET mutant pheochromocytomas. And this suggests the potential utility of kinase inhibitors against these tumors, which leads me to my significance. So TMEM mutant pheochromocytomas could be sensitive to RET selective inhibitors. These RET selective inhibitors are actually currently clinically available drugs that could be applied to these tumors. Additionally, the kind of full scope of TMEM-driven cancers isn't completely known. So this mechanism could be applicable across other cancers, such as renal cell carcinomas. TMEM mutations have been seen in those, and also increased RET expression has been seen. So that mechanism in those diseases as well. And this, again, further beyond TMEM, this just emphasizes the importance of RET localization within the cell to its transforming ability. So as far as next steps, I kind of mentioned this when we were talking about clathrin-mediated endocytosis. So I'd like to further explore some protein interactions in this process to kind of establish at what stage this effect is occurring. And one way we can do this is called proximity ligation assay. It's kind of similar in concept to a co-IP where you're identifying interactions. However, this is it's more sensitive to direct interactions. So any time you get a direct interaction, you get a red dot. So in the absence, I did a test in red cells. In the absence of GDNF, we get actually no dots. And as soon as you add GDNF, the RET and AP2 interaction is haywire. So I want to see if that is different in TMEM knockout cells. If you get less AP2 interaction, that why it's not being recruited. And there's several other interactions I'd like to look at in that area. Additionally, going along with the kind of change in membrane dynamics, we could also look at changes in membrane structure, potentially at microdomain organizations. And then kind of moving beyond TMEM and just looking at RET, I'd eventually like to just investigate the importance of RET mutant trafficking and its effects on cellular functions. So I'd like to thank my supervisor, obviously, and my committee members, Dr. Chan and Dr. Crocker, as well as my collaborators, Dr. Costin Antonescu, all the turf microscopy in Toronto, as well as Patricia Dahlia from the University of Texas Health Science Center, I think, and then my current lab members. Questions?
Questions for Tim? Or online?
Yes. Oh, Peter's. Okay, go ahead, Peter. You're muted if you're talking.
Right. Now I'm unmuted.
That'll
Sorry
help.
about that.
Okay.
It's a great talk, Tim. Congratulations on that really interesting story. This work has come along an enormous amount over the years that people in your lab have been studying this protein. I'm curious to see. So you're showing this lovely change in the dynamics of clathrin-coated pit formation. Do you know anything more about what TMEM 127 is associating with and how it's mediating these effects?
Short answer is no. There's some people who think that TMEM may be interacting with kind of ubiquitination proteins, but I don't really know how that would alter membrane dynamics. Yeah, we're not really sure.
Okay.
Yeah.
That's his next project.
Yeah.
Yeah, okay. I'll look forward to that. Thanks.
So, congratulations. There's a lot to take in when you stuff it.
Yeah.
Fire.
Yeah, that's why I like diagrams.
So these two proteins are expressed on the same cell soon, is that correct?
For the most part, yeah.
So
Yeah.
and you say that red actually is so what happens with TMEM? Is that also switched off? Does that continue to be expressed?
I think it continues to be expressed. Yeah, we don't really know its function other than what I presented. But yeah, I think it's just kind of always there.
Show them something. TMEM mutations on trafficking to the cell membrane. But you show the TMEM side effects.
Yeah.
Then the rest of your study, as I understand it, requires TMEM to be there to interact there. Does that occur at the cell surface?
Well, we don't actually think that RET and TMEM actually interact, like physically. I think RET is just.
Where does the mutant TMEM end up interact? So how does it affect organization?
We think that by mutating TMEM, it's removed from the membrane. So whatever it's doing within the membrane is lost. So if it's stabilizing CCPs, you remove it, the CCPs aren't stable, and they can't then bring RET into the cell. So we don't think it's a direct kind of interaction. The effect on RET is kind of a byproduct of TMEM getting removed.
So last question. These plasma gene regions. So do you know, suspect that to these cells are they more heterogeneous or happens about?
Yeah, that's something we're curious about. Like if the membrane is all kind of screwed up, maybe the lipid rafts are smaller, maybe they're, don't really know. So no, they look virtually the same. They're a little bit more spindly, but I think that might be due to kind of the overexpression of RET, or not simulate expression.
So actually, some precise possibility.
Yeah. So,
Shalom.
Yeah, related. A lot of data. Sorry. Yeah, I also have tried to just knockout. Then you see the it's also find that this is also a good chat. Like VGF, we studied many other. So did you check this receptors?
well,
See whether they also handle specific.
yeah. Well, with the turf, when I tried EGFR, it looked like it was the same. Yeah, there are other receptor types and kinases expressed in these cells. They're a little bit lower expressed. But yeah, I think it would be worth seeing if same.
Next question. Knockout. Right. See whether if you knock out timber.
Yeah.
If you knock out using inhibitor knock. So see what
No, we didn't.
treat other see whether also
Yeah. No, I used that one select because the constitutive activity is RET mediated. So I
think
don't.
specify specific.
Right. Okay. Yeah. No, we haven't.
Also,
Yeah. I only use that on the signaling, but yeah. Yeah.
with speculative. There are many things that get in. So I wonder some of them